Hello students, welcome back to my channel. We have completed the second chapter, that is sexual reproduction in flowering plants, and I have uploaded all the videos of the chapter sexual reproduction in flowering plants in my YouTube channel. You can watch the videos and you can clarify your doubts. Now we are going to discuss about the MCQs regarding the chapter sexual reproduction in flowering plants. Look at the first question, students. The initiation and development of floral primordium takes place by. Actually, we will call the flower as a modified shoot. I mean, the shoot gets modified into flower. And this floral primordium will be developed in the region of shoot apex when the plant has transferred from vegetative phase to reproductive phase. In the vegetative phase, only growth will be occurring when the plant will be transmitted from vegetative phase to reproductive phase. At that time, there will be a insertion of floral primordium, and that floral primordium will be initiated with the help of two factors. One is hormonal changes as well as structural changes in the plant. That's why here the correct option will be option D, that is both by hormonal and structural changes in plant. And the next question, oral of stamens in a flower represents. Let us recall the parts of the flower. You know that the outermost oral of the flower is called as a calyx, and each individual unit of calyx is called as a sepals, and the next oral is called as a corolla. Each individual unit of corolla is called as a petal, and the next oral is called as androecium. Androecium is the male reproductive part of the flower, and androecium comprising of many individual units called as stamens. So we can represent a stamen like this. So this is a typical stamen comprising of two parts. So this is the anther, and this one is the filament. Together it is called as a stamen, and many number of stamens comprising of the androecium. And the next oral will be the gynoecium, and gynoecium usually comprising of carpels. So here the question is oral of stamens in flower represents. So all the orals of stamens are together, it is called as androecium. And the next question, oral of carpels in a flower represents. Already I told you. So a oral of carpels in a flower represents the gynoecium. So gynoecium, it is the female reproductive part of the flower comprising of many number of carpels. And the next question will be, so the next question, what indicates A in the below figure? You know that this is the structure of anther, actually a matured anther which undergo dehiscence. So here they have labeled it as A. So what the A represents in this diagram? Pollen grains. You know that actually anther comprising of four wall layers. The outermost layer will be the epidermis, next followed by endothecium, then middle layer, then tapetum, and tapetum is the innermost layer. Within the tapetum, we can find the many number of microspores. Later, microspores will be developing into pollen grains. As it is a matured anther which undergo dehiscence, now these cannot be called as microspores. Instead, they are called as pollen grains. So here the correct answer will be the pollen grains. And the next question. So next question, oral of petals in a flower represents. So before only I told you the many number of petals are oral of petals in a flower represents the corolla. Corolla, these are the brightly colored petals. All the petals together it is called as corolla. And next question, the oral of sepals in a flower represents. So oral of sepals in a flower represents the calyx. Calyx, it is the outermost layer of the flower which gives protection to the flower when it is in bud condition, and each individual unit of calyx is called as a sepal. So the correct answer is calyx. And next question, stamens consist of which of the following parts? So I told you before only, a stamen, a typical stamen will be looks like this. It is comprising of an anther and a filament. Together it is called as a stamen. So now the question will be, stamens consist of which of the following parts? Option A, filament, option B, style stigma, C, anther, and option D, both A and C. So it comprising of filament as well as anther. And the next question, the number and length of stamens in flowers are. So number and length of the stamens in flowers are, they are variable in different species, because you know that there are different species of flowers comprising of different types of stamens, and the length of the stamens varies from one species to another species. Even the number of stamens also varies from species to species, because the number of stamens may ranges from one to many. In one species we can find one stamen, in another one two stamens. Likewise, there is always we can find the variation in the number of stamens. Even the length also greatly varies in different species. So here the correct answer will be, they are variable in different species. Next question, a typical angiosperm anther is unilobed and dithecous, bilobed and dithecous, maybe both A and B, and D bilobed and tetrathecous. We all know that a typical angiosperm anther is a bilobed anther and dithecous. So this one, so actually we will draw a typical anther like this. So here we can find two lobes. So this is one lobe, this is one lobe. That's why it is called as bilobed anther or dithecous. So theca refers to the chambers or lobes. That's why a typical angiosperm anther is always a bilobed and dithecous anther. And next question, the anther in transverse section appears to be, when we take a transverse section of this anther, when we take a transverse section of the anther, and this anther appears to be like tetragonal. So it appears like a tetragonal in appearance. When we take the transverse section of the anther, it looks like a tetragonal in nature. And next question, so here you can look at the 16th question. What are A, B, C and D in this figure? See here, this is actually the gynoecium part they have given. Here A, you know that A represents the stigma, and B is the style, C is the ovary, and D is the thalamus part where the gynoecium will be situated. So A is stigma, style, ovary and thalamus. We'll see which one will be the correct answer. So here they have given four options. In that, option C, it is the correct answer, because A is the stigma, B is the style, C is the ovary and D is the thalamus part. And next question, tetragonal anther consists of. So already I told you that the anther will be, when we take a transverse section, it will be tetragonal in nature. So that tetragonal anther consists of how many microsporangia? So this is the tetragonal anther. So the tetragonal anther comprising of 4 microsporangia, because this is one lobe, this is one lobe. It is a bilobed anther or dithecous anther, bilobed or dithecous anther. And it usually see, for example, so this is the bilobed anther, and this bilobed anther usually comprising of 4 microsporangia. It usually comprising of 4 microsporangia. So the correct answer will be 4 microsporangia. Next question, how many microsporangia are there in each lobe of anther? Actually this is one lobe, this is one lobe, this is one lobe. In one lobe we can find 2 microsporangia. Altogether it comprising of 4 microsporangia. But here they have asked how many microsporangia are there in each lobe of anther. So this is one lobe and this is one lobe. So each lobe comprising of only 2 microsporangia. And next question, pollen sacs develops from. So actually the microsporangia are only called as a pollen sacs because these microsporangia comprising of microspores, later the microspores develops into pollen grains. That's why the microsporangia is only called as a pollen sac. So pollen sacs develop from the microsporangia. And next question, typical microsporangium appears dash in transverse section. So we know that the typical anther appears to be tetragonal in outline, but they are asking about this microsporangia. The microsporangia, typical microsporangium appears to be circular in outline because each microsporangium appears like a circular in outline. And next question. Which one among us the given perform the function of production in typical into microsporangium? Which one among the given perform the function of production in typical microsporangium? So here they have given the wall layers of the anther. Option A epidermis, option B endothecium, option C is tapetum, option D middle layer. You know that tapetum, it is the nutritive layer which always provides nourishment for the developing microspores or pollen grains. Then remaining three layers, they will perform the function of protection because the outermost epidermis, endothecium and middle layer, all these three layers will perform the function of protection for the microsporangium. So option A, B and D are the correct answers. Next. Which of the following layer of microsporangium provides nourishment to the developing anther? So the nourishment layer or the nutritive layer will be called as the tapetum. It is the innermost layer of the microsporangium which always provides nourishment for the developing microspores. So correct answer is tapetum. And next question. Function performed by the outer three layers of microsporangium. The outer three layers are called as epidermis, endothecium and middle layer. So production to developing pollen, provides nourishment to developing pollen, helps in the digestion of anther to release the pollen. Actually the nourishment will be provided by the innermost layer called as a tapetum. Therefore the outer three layers will perform the function of production to developing pollen and even they helps in digestion of the anther to release the pollen. When the anther matures, the pollen grains has to be released from the anther. At that time the three layers will cause the digestion of the anther to release the pollen. So that option D is the correct answer. And next question. See, identify the parts A to I in the given figure or in this figure. So they have been given labelings option A to I. We'll first look at the parts. What are the parts called as? Here option A, label A, it is the style part of the flower. And option B is the stigma. And C is called as anther. Next D is the petal. E is the filament. And F is the sepal. G is the, actually this is the nectariferous area where the nectar will be collected in certain flowers. And H is called as ovary. And H is the ovule. Actually they have been not labeled. H is called as ovule. Here they will be the ovules. And I part is called as the ovary. So these are the actual labelings they have given. They told to identify the parts from A to I in this figure. So when we look at the flower and when we look at the options, option D is the correct answer. When you go through the labelings, so option D will be the correct answer. A, it is the style. B, stigma. C, anther. D, petal. E, filament. F, sepal. G, the nectariferous area. H, ovule, and I is the ovary. And next question. The microsporangium cells which possess dense cytoplasm and have more than one nucleus is the characteristic of. When we look at the wall layers of the anther, the tapetum. So tapetum, this is the innermost layer, the nutritive layer, and the cells of the tapetum, they contain dense cytoplasm and many number of nuclei. So they comprising of more than one nuclei also. When we look at the cells of the cells of the tapetum, each cell comprising of dense cytoplasm and more than one nucleus. So that is the characteristic feature of tapetum. And next question. In young anther, the tissue occupying the center of each microsporangium is called. So in this anther, so this is the microsporangium. In young anther, the tissue occupying the center of each microsporangium. So in each microsporangium, there will be a tissue filled. That tissue is called as a sporogenous tissue. And that sporogenous tissue later gives rise to microspore mother cell, and microspore mother cell will develops into or will gives rise to the microspores or pollen grains. That's why, so here each microsporangium is filled with sporogenous tissue. And next question, identify A to E in this figure. So the diagram they have given, it is the typical embryo sac, the female gametophyte called as embryo sac. So this is a typical embryo sac. So here actually what they have labeled is A. So here you can observe the larger cells, these are towards the micropylar region, and these are the cells which are towards the cell region. Here A represents the synergids, and B is the egg, the larger one called as egg, the female gamete, and C is the central cell. So this entire thing is the central cell, and D, these two are called as polar nuclei, and E represents the antipodals which are at the cell end. So according to the diagram, there are labelings A to E, and these are being identified as A are the synergids, B is the egg, C is the central cell, D are the polar nuclei, two polar nuclei, and E is the antipodals. And when we look at the options, option A is the correct answer. Option A is the correct answer. Next question. Arrange the following layers of microsporangium according to their presence from inside to outside. So they are asking, so this is the microsporangium, this is the microsporangium, and here it is comprising of 4 wall layers. So they are asking to arrange the wall layers from inside to outside, asking to identify the wall layers from inside to outside. So this is the innermost layer called as a tapetum. So first should be tapetum, and the next layer is called as middle layer. The second one will be middle layer, and next layer will be endothecium. The third one is endothecium, and the outermost layer is called as epidermis. So here C, B, A and D. So that option D is the correct answer. Option D is the correct answer. So here option D, so option D is C, B, A and D. And next question, sporogenous tissue of microsporangia is option A, groups of compactly arranged homogeneous cells. Option B, occupies the center of microsporangia, and option C, present inside young anther, and option D, all of the above. So sporogenous tissue, it can be seen in the microsporangia, and that sporogenous tissue is actually the group of compactly arranged homogeneous cells, and they occupy the center of microsporangium also, and they are present in the inside the young anther. Because in the mature anther we cannot find the sporogenous tissue, we will find the pollen grains. So for this sporogenous tissue, here all are correct. So option D, all of the above. And next question, each cell of microspore tetrad is. So for example, if this is the microspore mother cell, you know that mother cells are always diploid. They undergo meiotic division, and after meiotic division it will form a tetrad. So this is a microspore tetrad. So as these microspores are formed by the meiotic division, all the cells becomes haploid. So each cell of microspore tetrad will be haploid in nature because they are formed as a result of meiotic division from the microspore mother cell. So that the correct answer will be haploid. Then the next question, Which type of cell division occurs in the cells of sporogenous tissue to form microspore? So already I told you it is a meiotic division. So even the sporogenous tissue, it will undergo meiotic division. Then only the microspores will be produced. So that is called as reductional division or meiotic division. And next question. Formation of pollen from pollen mother cell is referred to as. Formation of pollen from pollen mother cell are also called as microspore mother cell is called as microsporogenesis. So we will define the microsporogenesis like that only. The formation of microspores or pollen grains from microspore mother cell or pollen mother cell is termed as microsporogenesis. That's why the correct answer will be microsporogenesis. Next question. Pollen grains are option A microspore tetrad, option B dehydrated microspores, option C megaspore tetrad, option D pollen mother cell. Actually the pollen grains are said to be the dehydrated microspores because the microspores later develops into pollen grains. At that time they lose a certain amount of water. That's why we will call it as a dehydrated microspores. And next question, male gametophyte in angiosperm is represented by. The male gametophyte in flowering plant is said to be the pollen grain and the female gametophyte is said to be the embryo sac. So here they are asking about the male gametophyte. The male gametophyte will be the pollen grain. And next question, pollen grains are spherical, oval, generally spherical, irregular. So we cannot see the exact shape of the pollen grains. They may be spherical, they may be oval, they may be irregular, but mostly, in most of the species we will find spherical shape. That's why we will say generally spherical. And next question, diameter of pollen grain is. A typical pollen grain ranges from measures about 25 to 50 micrometers in diameter. And next question, the exine of pollen is. Option A, hard outer layer of pollen grain. Of course it is a hard layer. And B, most resistant organic matter noun. Yes, it is the most resistant material. And option C, layer made up of sporopollenin. Yes, exine is always made up of sporopollenin, a highly resistant organic chemical. And option D, layer which can withstand high temperature, strong acids and alkali. Even this is also correct. And option E, layer which cannot be degraded by any known enzyme. Yes, there is no enzyme to degrade the exine layer because of the highly resistant organic chemical called as sporopollenin. Which of the following is correct regarding this one means here all are correct regarding the exine. So you can go through all the options, all are exactly relevant to the exine. So these are the certain questions which we can discuss today. Remaining questions we can discuss later in the next class. Thank you, students.